What if I told you that the only fundamental difference between a really soft, digestible slice of bread and like a completely indigestible block of solid wood is just the microscopic angle of a single chemical bond?
I mean, honestly, it sounds impossible because
Yeah.
you look at bread, you look at wood, and they just seem like, well, entirely different universes of matter.
Right.
But chemically,
Yeah.
they are literally constructed from the exact same microscopic building blocks.
Which is wild. And that's exactly why today we're doing a deep dive into those literal building blocks of, well, every living cell in your body. We're looking at macromolecules.
Yeah, the big ones. Carbohydrates, lipids, proteins, and nucleic acids.
Exactly. And our mission today is to take what is usually this really rigorous, highly technical study topic.
Especially if you're prepping for nursing entrance exams like the TEAS or the
Yeah.
HESI.
Oh, for sure. We want to unpack this for those test takers, but also just for anyone listening right now who is insanely curious about how life actually works. Because, I mean, whether you're entering the medical field or you're literally just eating a sandwich, these molecules are running your life.
They really are. And I know anyone staring down a massive biology textbook right now probably feels a bit overwhelmed by the sheer volume of vocabulary.
Oh, total. It's basically a foreign language.
Exactly. So we
Yeah.
really want to focus on how these things actually operate in reality.
Okay, before we get into how a microscopic angle turns bread into wood, which I'm very excited about, I understand there's actually a bit of a biological scandal with that big four list I just read off.
Oh yeah, there is. If you ask a strict biochemist about carbs, lipids, proteins, and nucleic acids, they'll tell you that the actual definition of a macromolecule is highly contested.
Wait, really?
Yeah, one of the big four is technically an imposter.
An imposter. In
Yeah.
the foundational textbook list. Okay, I definitely want to get to that. But I guess to understand why one doesn't belong, we probably need to know how scientists even figured out these things existed in the first place, right?
Right. And to do that, we have to rewind to the early 1920s
Okay.
because at that time, the greatest scientific minds on the planet, I mean, established, highly respected chemists,
Yeah.
they didn't even believe giant biological molecules existed at all.
Wait, so they were studying things like starch and rubber, right?
Yeah.
But they didn't think they were molecules.
Exactly. They called them colloidal aggregates.
Colloidal aggregates. Get you.
Yeah. The prevailing theory was that these substances were basically just loose clusters of small, ordinary-sized molecules.
Okay, so like a cluster.
Yeah, think about what happens when you drop liquid soap into water. The soap molecules naturally clump together because of these weak physical forces, but they aren't physically attached to each other.
Oh, I see. So the scientific establishment basically pictured these biological substances as well, like an unorganized flash mob.
The flash mob, yes.
Just a bunch of tiny molecules hanging out in the exact same area but not connected.
That perfectly captures the mindset.
Mhm.
But then, this German organic chemist named Hermann Staudinger comes along in 1922,
Okay.
and he proposes what he called the macromolecular hypothesis. He'd been studying rubber and basically argued that the whole flash mob theory was completely wrong.
Bold move in the 1920s.
Very. He said these were actually single molecules of just enormous size, made of thousands of atoms, connected end to end by ordinary, unbreakable covalent bonds.
So instead of a flash mob, I'm imagining like a giant physically connected freight train.
Exactly, a single massive train.
But I'm guessing established scientists didn't really love being told their fundamental understanding of chemistry was completely backwards.
Oh, the backlash was fierce.
Yeah.
I mean, the idea of a single molecule being that massive just seemed physically impossible to them. It was almost offensive to their understanding of nature.
Wow.
Yeah, at one lecture, a prominent colleague supposedly stood up and told Staudinger to just abandon the whole idea.
Just give it up.
Yeah, he claimed that a molecule of that size was as absurd as a zoologist reporting they had found a 100-meter-long elephant.
100-meter-long elephant.
Yeah.
I swear, the historical shade in the scientific community is always so aggressive.
It really is. But Staudinger just spent the next decade proving them wrong. He didn't just argue. He meticulously measured the molecular weight of these substances.
Like doing the actual math.
Right, looking at things like viscosity and osmotic pressure, until the data was just undeniable. And he finally won the Nobel Prize in Chemistry in 1953.
So he actually lived to see the ultimate victory.
He did. And this conceptual leap was vital, because if the scientific community hadn't accepted that giant, continuous molecules could exist, well, scientists later on could never have conceptualized DNA as one massive, continuous double helix molecule.
Oh, wow. That makes so much sense. Okay, so Staudinger proves the giant freight trains exist. But if our bodies are constantly, breaking down food and building up muscle, how are we managing these massive trains?
Good question.
Like, how do we actually couple and uncouple the individual cars?
So at their core, these macromolecules are governed by two universal, completely opposing chemical reactions.
Okay.
To build a train, your cells use a process called dehydration synthesis. Sometimes you'll see it called condensation.
Well, dehydration obviously implies losing water.
And that is the crucial mechanism. You take smaller, single units, those are called monomers.
Right.
And if you want to join them together to make a larger chain, a polymer, you have to forge a covalent bond between two monomers.
Okay. And how does the water fit in?
To form that bond, a water molecule literally has to be removed from the ends of those molecules. It gets released in the process.
Let me see if I have this visual right for everyone listening. It's almost like, like paying a toll on a highway.
I like that.
To get the gate to go up and officially link two train cars together, you have to toss a coin in the bucket. But in this case, the toll is a single water molecule. You drop the water, the bond forms.
That analogy actually holds up really well. And obviously, the body needs a way to reverse that process, right? When it's time to break those train cars apart, it uses the exact opposite reaction, which is hydrolysis.
Hydrolysis.
Yeah. Hydro means water, and lysis means to break or split.
So the toll booth essentially gives the water back, and the bond breaks.
Precisely. To sever that covalent bond, a water molecule is forced back into the structure, splitting the large polymer back into separate monomers.
Okay.
And for anyone listening right now, you actually experience massive scale hydrolysis every single day.
Wait, really? What?
It is literally what digestion is.
Oh.
Yeah. When you eat a complex carbohydrate, your digestive system uses water and specialized enzymes to snap all those chemical bonds, breaking the food down into tiny monomers that are small enough to actually cross into your bloodstream.
Okay, that makes the abstract chemistry feel very real. So let's look at the actual macromolecules that follow this repeating train car rule, the true polymers. Let's start with carbohydrates, since you just mentioned eating them.
Sounds good.
Yeah.
So carbohydrates are built from monomers called monosaccharides.
Saccharide just means sugar, right?
Exactly.
Right.
These are your simple sugars, glucose, fructose, galactose. And they actually all share the exact same chemical formula, which is 6 carbons, 12 hydrogens, and 6 oxygens.
Okay.
And when your body pays the water toll and links 2 of them together via dehydration synthesis, you get a disaccharide.
Like sucrose.
Yep. Sucrose, which is just regular table sugar, is just a glucose and a fructose linked together.
Oh, okay.
Lactose, the sugar in milk, that's a glucose linked to a galactose. And when you link thousands of them together, you get these massive polysaccharides like starch of potatoes or glycogen in human muscles.
Ah, glycogen. The sources mention glycogen specifically in the context of marathon runners.
Yes, the classic example.
And you see a runner hit the wall and physically collapse at mile 20. That isn't just a mental barrier, is it? It's literally a biochemical depletion event.
It is an absolute depletion because an average adult stores a very limited amount of glycogen, mostly in the liver and skeletal muscles.
So it's like a backup tank.
Right. It acts as an easily accessible energy reserve. But when an endurance athlete burns through that stored polysaccharide macromolecule, they simply run out of fuel. That physical wall they hit is the exact moment the body's glycogen tanks hit absolute zero.
Wow. Okay, so this brings me back to the question I asked at the very beginning of the deep dive, the bread versus the wood.
Yeah.
The sources pointed out they are both made of pure glucose monomers, just giant chains of glucose. So how can one give me energy to run a marathon and the other wood does absolutely nothing for me?
It all comes down to geometry. The only difference is the physical geometry of the chemical bond linking those glucose monomers together.
It's just the angle.
Just the angle. In starch, the glucose molecules are linked by what we call an alpha-1-well-4 glycosidic bond. They lock together in a very specific angle.
Okay.
But in cellulose, which is the structural material of wood and plants, they lock together with a beta-1-0-4 glycosidic bond. The angle is slightly different.
And that tiny shift in angle literally changes everything.
It really does.
Yeah.
Because human digestive enzymes are essentially like highly specific physical keys.
Oh, so they have to fit into a lock.
Exactly. They are perfectly shaped to fit the lock of the alpha bond in starch. They just slip right in, perform hydrolysis, and break it down for energy.
But with cellulose.
Those same enzymes physically cannot fit into the geometric shape of the beta bond in cellulose. It just doesn't fit.
So the wood or the plant material just passes right through our system untouched.
Exactly. We just call it dietary fiber. It is the exact same monomer, the exact same chemical formula, but a radically different biological outcome purely dictated by geometry.
That is wild. Geometry literally dictates destiny.
It really does.
So if carbohydrates are mainly about storing and releasing energy, what about the machinery that actually does the work? The enzymes, the muscles.
That brings us to the second true polymer, proteins. They are basically the workhorses of the cell.
And what are their train cars?
Their monomers are called amino acids. There are 20 standard amino acids that string together to form polypeptide chains. But with proteins, a straight chain isn't enough to actually do any work.
What do you mean?
It is entirely about how that chain folds up into a three-dimensional shape.
How complex does the folding really get, though?
Oh, incredibly complex. There are 4 distinct levels of structure.
Oh, got me through them.
So the primary structure is just the straight sequence of amino acids, just the order of the train cars.
Got it.
The secondary structure forms when that chain starts naturally twisting into spirals or folding into these pleated sheets just based on local chemical attractions between the amino acids.
Okay, so it starts crumpling up a bit.
Right. Then the tertiary structure is the overall highly complex 3D shape of that single entire chain.
And the fourth?
Finally, the quaternary structure occurs when multiple separate folded chains actually lock together into a massive superstructure.
So the body is taking these strings, curling them, folding them up, and then snapping different folded blocks together to build a microscopic machine.
Yes. A perfect example of this is hemoglobin, which is the protein responsible for carrying oxygen inside your red blood cells.
Right. Everyone's heard of hemoglobin.
Right. It has a quaternary structure made of 4 distinct polypeptide subunits locked together. And this actually highlights an incredible yet devastating example of why the exact sequence of those initial train cars matters so much.
The sources talked about sickle cell anemia in this context, right? Back in the 1950s, a scientist named Vernon Ingram figured out the exact chemical cause of the disease. What did he actually find?
He discovered a tiny, tiny error in the sequence. Out of the 146 amino acids that make up just the beta chain of hemoglobin, just one single amino acid is wrong in sickle cell anemia.
One typo out of 146.
Just one. One specific glutamic acid is accidentally swapped out for a different amino acid called valine.
That's almost nothing.
But that single substitution completely changes the entire 3D shape of the hemoglobin protein.
Because the fold gets messed up.
Exactly. Under low oxygen conditions, that altered shape makes the protein stick together in these long, rigid fibers.
Oh, wow.
And this physically distorts the red blood cells, pulling them into a sickle shape, which then gets stuck in blood vessels and cause all the symptoms of the disease.
It's terrifying to think about the sheer uncompromising precision of these macromolecules. A single wrong link in a massive chain changes the entire 3D shape and causes a life-threatening disease.
It really is staggering.
Which naturally leads me to my next question. If proteins require such insane precision, how does the body actually know the correct sequence? How does it avoid making that single typo?
Well, it needs a blueprint. And that is the job of the third true polymer, nucleic acids, DNA and RNA.
The blueprints. Okay, and what are their train cars?
Here, the monomers are called nucleotides.
And what does a nucleotide look like compared to, a simple sugar or an amino acid?
They're a bit more complex. Every nucleotide has 3 components: a 5-carbon sugar, a phosphate group, and a nitrogenous base.
Okay, so it's a 3-part car.
Yeah. And DNA uses a sugar called deoxyribose, along with the bases adenine, thymine, guanine, and cytosine.
Right, the classic ATGC.
Exactly. RNA is slightly different, though. It uses ribose as its sugar and swaps out thymine for a base called uracil.
Now, the story of discovering the DNA structure is legendary, right? Watson and Crick publishing the double helix in 1953, heavily relying on Rosalind Franklin's famous X-ray diffraction image, photo 51.
Oh, absolutely. And Erwin Chargaff discovering the base pairing rules, that adenine always matches thymine.
But I didn't realize until reading our notes for this deep dive that there was actually a massive failure happening at the exact same time.
Yes. Linus Pauling.
But Linus Pauling.
Yeah, he was a brilliant chemist, actually the one who first described sickle cell anemia as a molecular disease. And he was also racing to solve the structure of DNA. In early 1953, Pauling actually published a model before Watson and Crick did.
Wait, really? What was his model?
He proposed that DNA was a triple helix.
A triple helix. But our notes say he made a fundamental chemistry error with how he physically built it.
He did. It was a massive oversight. He placed the phosphate backbone on the inside of the helix with the nitrogenous bases pointing outward.
Wait. If the phosphate groups are on the inside, tightly packed together in the core, aren't they negatively charged?
They carry a very strong negative
Yes. So
charge.
if you cram all those negative charges into the tight center core of a molecule, wouldn't they just violently repel each other?
Yes, the molecule would literally throw itself apart.
So one of the greatest chemists in the world published a model that basically ignored basic magnetic repulsion.
It was an astonishing oversight, especially from someone who had essentially written the textbook on chemical bonds.
That is crazy.
When Watson and Crick saw Pauling's paper, they instantly realized his model was physically impossible. And it actually spurred them to rush their own correct double helix model to publication.
Wow. It serves as a huge cautionary tale, honestly. Even the most brilliant minds fail when they ignore the fundamental geometry and physics of the molecules.
Absolutely.
All right, so we've got carbohydrates, proteins, nucleic acids. They all follow the rules. They are true polymers, monomers linking together endlessly, paying the water toll to couple the cars.
Right?
But you mentioned an imposter at the top of the show. We have to talk about lipids.
Yes. Lipids are the absolute rule breakers. This group includes your triglycerides, which are fats and oils used for long-term energy.
Okay.
It includes phospholipids, which build cell membranes, and it includes steroids, like cholesterol, estrogen, and testosterone.
But why are they imposters? I mean, they are huge molecules. They are organic.
True, but they do not polymerize.
Oh, they don't form chains.
Exactly. Recall our strict definition of a true macromolecule. A massive chain of identical or similar monomers strung together indefinitely. A lipid, like a triglyceride, just isn't a chain.
What is it then?
It is simply a glycerol backbone with exactly 3 fatty acid tails bolted onto it. And it doesn't repeat. It stops right there.
So calling lipids macromolecules is basically a technicality.
Pretty much.
They don't fit the strict definition of a repeating polymer train, but I'm guessing they're just clearly too important to leave off the list.
Exactly. It's essentially a biological grandfather clause. Because without lipids, life as we know it simply doesn't exist.
Really? Why?
Consider phospholipids. Their chemical structure gives them a highly unique property. One end, the head, is hydrophilic, meaning it loves water.
Okay.
But the other end, the fatty acid tails, is hydrophobic, meaning it actively repels water.
Okay, so how does that actually help build a cell?
Well, in a watery environment like the human body, phospholipids naturally arrange themselves tail to tail.
To hide from the water.
Exactly. The water-fearing tails hide on the inside of this double layer, while the water-loving heads face outward toward the environment. In 1972, scientists proposed the fluid mosaic model to describe this.
And that double layer becomes a membrane.
Yes. This delicate, self-assembling lipid bilayer forms the physical boundary of literally every single cell in your body.
Okay, so if they are literally holding our cells together and acting as the border patrol, I guess we can let them stay in the macromolecule club.
I think we have to.
But since we're talking about lipids, I do want to tackle some of the bad PR they get, because it feels like the general public fundamentally misunderstands a lot of these molecules, especially when it comes to diet.
Oh, completely. Nutrition is where the actual science often gets very distorted. The most pervasive misconception by far is that all fats, all lipids are inherently unhealthy and should just be avoided completely.
Right. I remember the whole low-fat craze of the 80s and 90s. Like, everything in the grocery store suddenly had a bright green fat-free sticker on it.
Yeah, and that was heavily driven by research from people like physiologist Ansel Keys, who linked dietary fat to heart disease. But the biological reality is incredibly nuanced. Lipids are strictly essential. Your body literally cannot synthesize certain fatty acids, like omega-3 and omega-6. You must get them from your diet.
Oh, so you have to eat fat.
Yes, and you absolutely need cholesterol to synthesize vital hormones. The danger was never lipids as an entire category. It was always about the specific types, like artificially hydrogenated trans fats and the overall quantities.
That makes total sense. And honestly, carbohydrates suffer from the exact same generalization. People just hear carbs are bad, but as we covered, they aren't interchangeable at all.
Not at all.
The simple sugar in a can of soda is handled completely differently by your biology than the complex cellulose fiber in a piece of broccoli, even though they're both technically carbohydrates.
Exactly. And there's actually another crucial misconception, this time involving nucleic acids.
Oh, what's that?
Well, we constantly talk about DNA and RNA as genetic storage, right? Just blueprints.
Right.
But nucleotides actually have a completely different, massive job in the body. Adenosine triphosphate, or ATP, is the main energy currency of the entire cell.
Wait, ATP is a nucleotide.
Yes, ATP is literally a modified nucleotide. So nucleic acids aren't just for heredity, they physically power your metabolism.
Wow, I never made that connection.
Most people don't.
Okay, well, if we're talking about shattering biological assumptions, we absolutely have to bring up the ultimate rule breaker from our sources. We mentioned sickle cell being a protein folding error, but the notes describe something called a prion, which honestly sounds like something out of a horror movie.
It kind of is. Prions force biologists to rewrite the textbooks entirely.
Really?
Why? To understand why, you first need to understand the central dogma of molecular biology. For decades, it was just an absolute, unquestioned assumption that hereditary transmissible information could only be carried by nucleic acids, DNA, and RNA.
Right, so if a virus or a bacteria is infectious and replicating, it has to have genetic material to give instructions on how to replicate.
Exactly. But in the 1980s, a scientist named Stanley Prusiner was studying these terrifying neurodegenerative diseases.
Like what?
Things like bovine spongiform encephalopathy, better known as mad cow disease, and Creutzfeldt-Jakob disease in humans.
Okay.
And after exhaustive research, he perplexed something that sounded like pure science fiction. He claimed these diseases were being transmitted by a rogue protein.
Just a protein. No DNA or RNA involved at all.
None whatsoever. He discovered that a perfectly normal protein in the brain could just randomly become misfolded into an abnormal shape. He called this infectious protein a prion.
But wait, how does a protein with absolutely no genetic code multiply and infect an entire brain?
This is the terrifying part.
Yeah.
When this misfolded prion bumps into a normal version of the exact same protein, its physical shape somehow forces the normal protein to misfold into the rogue shape.
Oh my God, like falling dominoes. One goes rogue, touches the next one, and physically forces it to flip.
Precisely. It just cascades through the brain, leaving massive tissue destruction in its wake, purely through physical geometric shapeshifting.
That is so deeply unsettling.
It is. And much like Staudinger back in the 1920s, Prusiner faced immense skepticism. I mean, the scientific establishment thought it was completely impossible to have an infectious agent without genetic material.
But he proved them wrong.
He did. The data won out, and he was awarded the Nobel Prize in Physiology or Medicine in 1997.
Wow. Okay, so for you listening right now, you know the origin story of the freight trains. You know the true polymers. You know the lipid imposter. And now you know about the rule-breaking prions. But I want to tie all this directly back to practical reality. Why is this the holy grail of biology, and especially for people taking nursing entrance exams?
Because this isn't just abstract chemistry. It really is the foundation of everyday clinical practice. I mean, every nutrition label you read is literally a direct report on macromolecules.
Right, grams of carbs, fats, and proteins.
Exactly. And so many medical conditions are just macromolecule dysfunctions. Take lactose intolerance. A lot of people think it's an allergy. It isn't. It is simply the body failing to produce lactase, which is the exact enzyme needed to perform hydrolysis on the disaccharide lactose.
Oh, so the toll booth is closed, the bond can't be broken.
And gut bacteria ferment the sugar instead, causing all the symptoms.
That is so simple when you break it down like that.
Yeah.
Or what about phenylketonuria, PKU? The sources describe it as an inability to process a specific amino acid, phenylalanine.
Right. It's just a protein processing error at the molecular level.
Even the mRNA vaccines that became household names recently, that is literally the central dogma in action, right?
Yeah.
Delivering a nucleic acid into your cells to instruct them to manufacture a specific folded protein to trigger an immune response.
Exactly. For anyone entering healthcare, this is the underlying language of every patient's chart. When you read a blood glucose level, you are assessing carbohydrate metabolism.
Right.
A cholesterol panel is lipid transport. Checking hemoglobin A1C is literally measuring glucose molecules that have become physically attached to a protein molecule.
So if you're out there studying for the TEAS or the HESI, you really aren't just memorizing trivial definitions to pass a test. You are learning the vocabulary of human survival.
Couldn't have said it better.
So let's do a quick recap of the vital takeaways before we wrap. 1, the big 4 are carbohydrates, lipids, proteins, and nucleic acids.
Yeah.
2, they are universally built by dehydration synthesis, paying the water toll, and broken down by hydrolysis.
Exactly.
3, lipids are the odd one out, non-polymers. 4, structure dictates function.
And 5, prions prove that infectious information can be transmitted by shape alone without any genetic material.
It constantly comes back to shape, which actually brings me to a final thought I want you, the listener, to mull over.
Hmm.
When we think of biological information, we are trained to think of a written code, right? We picture the sequence of A, T, C, and G in our DNA basically acting like a printed software program.
Right, the blueprint.
But looking at all of this from the microscopic bond angles that determine if we can digest food, all the way to the infectious geometry of a prion biological information is just as often a physical, three-dimensional geometry. So if a molecule's folded shape can act as its own kind of biological instruction manual, passing on commands purely by how it physically bends and locks into place, what other physical geometries might be hiding biological secrets we haven't even thought to decode yet? How much of life's software is actually hiding in the shape of the hardware? Keep questioning, keep studying, and we'll catch you on the next deep dive.